Hello, everyone. My name is Nagarjun Kasaraneni. So, pretty much I work as like a scientific consultant right now, and for a seed startup. And for today's talk, I'm going to talk about like characterization of mRNA therapeutics using liquid chromatography based methods. I think the image here really shows the complexity of the drugs we have been like developed. They initially a small molecule, and then you go to a drug antibody-drug conjugate. From there there's a peptide, and then you move into like more like a complex recombinant protein, and then you have an antibody, and then you have this viral vector and a cell therapy. So it just shows like how we have advanced in biologics from a small molecule. So right now I'm going to talk more on the mRNA therapeutics, which are like which is a combination of a more like a small molecule with a lipid particle, which becomes like a more cell and gene therapy product. So this is a confidential statement. So I'm just making this talk based on the literature which has been published, available in public. No confidential information has been shared. It's more regarding like an educational talk, and these are mostly the views of the current literature. And yeah, just to make to really confidently say that like there's no confidential information in this. So for today's talk, the overview of the talk would be like I'm going to talk a little bit about the mRNA therapeutics and the cell and gene therapy products available, and really talk about like analytical method development in a drug product life cycle, and then talk about like introduce you to critical quality attributes, and then from there we will really deep dive into more towards like an LC methods that really measure like the RNA itself and the RNA therapeutic actually. So RNA therapeutics include like RNA plus like a lipid nanoparticle. So I'm more concentrated towards like an RNA molecule itself today, and talk about the CQS for the RNA molecule. And I'll give my final thoughts. So really looking at a history of like mRNA therapeutic landscape here, it all started around like 1961, and in 2020 we got like more of these approved for to combat infectious diseases. But the most important milestones across this whole life cycle is kind of the first cationic lipid delivery of mRNA happened in 1989, and then we kind of like have this lipid nano entrapment also happened in 1978. So the first time where mRNA has been like trapped into a liposome for a delivery. And in 2020 we got approval for two vaccines, which is kind of from Moderna as well as like Pfizer for COVID SARS-CoV-2 pandemic. So moving on, uh, I want to look into like more on like what are the approved cell and gene therapy products and the mRNA vaccine products available. So the currently like for cell and gene therapy products, we have like the gene edited product is the Casgevy and the Lyfgenia, which is for like sickle cell diseases. Casgevy is from Vertex, Lyfgenia is from like Bluebird. And we also have like an AAV gene therapy vector, which is kind of more towards like Eldevis for the intestinal muscular dystrophy. And Kymriah is one thing, which is a CAR T therapy for leukemia and lymphoma. And there are other gene therapy vectors that have been previously approved for Luxturna, Rotovac, Skyzona, and these are gene therapy as well as like we also have like these cell therapy vectors, which is Casgevy and Zizanma for gene therapy for beta thalassemia. And coming back, looking at the talk really focuses on mRNA therapeutics. I would say like mRNA vaccines, which is the Spikevax, and the Comirnaty, which is from like Pfizer and Moderna, which have been like utilized to really during the COVID-19 pandemic. Um, so based on all these approvals from cell gene therapy and the mRNA vaccines, it's predicted that like there will be 30 to 60 gene and cell therapy products available by 2030, and it's expected to see that like it's more than um, 350k more than the patients will be treated with the cell and gene therapy products. So before we move into like discussing on the CQAs for the mRNA, um, I would like to really talk about like how does this drug product life cycle happen from an analytical development point of view, and where does this mRNA um, manufacturing or mRNA vaccine really comes into play. So the first cycle we can really look at is like a drug development itself. We have a preclinical which you really work for to identify the lead candidate. Phase one, phase two are more like the safety, and safe three. Phase three is kind of more like a huge population. And then once you go for a BLA, you get an approval, and then you go for market. So that's just like a general life cycle for a drug. When you look at the same life cycle from kind of like um developmental stage, you can say that like preclinical phase one is still they consider to be early stage development. Once you go into phase two and phase three, it's called more like a late stage development, and once you have an approval and market, we call it a commercial. And looking at from more like a manufacturing point of view, so the early research and development phase, once we have this initial uh, process manufacturing, once the initial process development happens in R&D, and then we move into like during the phase one, phase two, we kind of like understand the process and stabilize the process. We call them engineering GMP. And once we have like more into phase three and approval, we have to establish this more um, clinical GMP manufacturing process, which is more robust, where you ideally do all the process characterizations, process PPQs, and to really make this process more established in a GMP facility. And then once you get an approval, you move it to commercial manufacturing, where it is like robustly validated, and it's been used to produce a commercial drug. And on the same lines, when you look at from an analytical development point of view, it's kind of like you have a defined design evaluate, which is more towards that initial evaluation of an assay. Once you kind of optimize the assay to a required thing, then you go into a qualification, where you ideally look at all the four parameters like linearity, specificity, um, accuracy, and robustness. And then you can move into validation, where validation is more towards the phase three, where you kind of test with different operators, different conditions, the assay is robust across different reagents, how the reagents play a role. You look at all these factors to validate the assay. Once it's validated, you have a commercial approval. We actually use that assay, like we kind of do a verification of that assay. So is this assay at different commercial manufacturing sites? Is this assay really uh, performing the same way as it's been validated? So looking the whole process from a mRNA mRNA therapeutic point of view, so ideally have like a linear pDNA, and you kind of create this whole bulk mRNA, and then which is called like a drug substance here, and then you kind of create this drug product when you mix it with the LNP. We call it like an mRNA therapeutic, and then that would be your final drug product. So to really establish the linear pDNA to an bulk mRNA, so we ideally can look at the process of manufacturing, how really it happens, and what are the reagents that we are required to create this whole mRNA molecule that is going to be utilized to actually fit into a lipid nanoparticle. So the synthesis of mRNA is normally through an in vitro transcription process. So the in vitro transcription process is just ideally a mixture of a T7 RNA polymerase with your nucleotides and a DNA template. So the DNA template ideally has your specific gene of interest, where your polymerase is going to generate the mRNA from, and you're going to provide with like different types of DNTP NTPs, and which are like modified and unmodified. You can ideally talk when we really talk about the molecule itself, how what's modified and unmodified. And you also have this pyrophosphate, and then once we kind of have this mixture, in vitro transcription mixture, and we ideally produce our mRNA molecule. So this process is a very kind of like an efficient process, but at the same time, you have a lot of components that to generate an mRNA. So whenever an mRNA is generated, it also brings a lot of impurities. It brings like an incomplete messenger RNA transcript. So whatever the full length you're going to generate, it cannot. Some of the molecules are not generated completely, and there's a possibility of like degradation of mRNA because mRNA is very sensitive to pH, temperature, other factors. And there's a possibility that like mRNA aggregates also form. Like once they form, they have an ability. It's a more like a negative molecule, like a phosphodiester bond is more negative. So this mRNA has this high ability to form secondary structures or interact with each other to form this double stranded aggregates. So these are all like impurities which ideally completely destroy our product of mRNA, which has to be like packed into our lipid nanoparticle. So understanding these impurities and really understanding the whole mRNA is a very important critical step. That's where we ideally look at all the critical quality attributes and which methods are really applied. So once we have this mRNA, we can really look at like how this mRNA is kind of like subdivided in such a way that like we can define our quality attributes. So the structure of a synthetic RNA can be defined as ideally into few different parts. The first part would be ideally we call the phi cap, which is, so you have a cap region and you have this triphosphate. And the phi cap is very important for ribosomal initiation, translation, and stability of this mRNA molecule. And then you have this 3-phosphate, and then you have this 5-UTR translation. So ideally the 5-UTR is like the region where you ideally help the polymerase to stick and really produce your mRNA. Use your mRNA to translation. So it helps for translation. So, and the coding region itself is the gene of interest you ideally put it in. So there are a few modifications people have identified across like developing this mRNA vaccines. So one of the modifications people regularly use is like to use this methyl pseudouridine and methyl pseudouridine. So the reason why people use this is kind of it has observed to show that like it reduces immunogenicity and enhances stability of the mRNA molecule. And the 3-UTR is the region where ideally you where your coding sequence and then you have a poly A. So poly A is also a very important part of this mRNA molecule where ideally it kind of like helps in the stability of this molecule when it really is delivered to a cytosol. And the cap and both poly A plays a very big role, and also in translation, ending the whole translational activity to produce the protein. So the molecule itself comes with a few different regions which ideally have to be identified, and those are when we talk about more on the LC methods, we'll understand like why each region is so important, what are the methods we ideally use them for. So completely understanding this synthetic RNA, I want to really talk more about like the CQAs before I introduce the different CQAs for this mRNA, synthetic RNA. I would like to talk about more on like how are these FDA guidance for critical quality attributes defined. So a critical quality attribute can be defined as a quality attribute that must be within an appropriate range, limit, and the distribution to ensure that the product quality is defined. So to define any of these critical quality attributes, we have a process. So the process, it's like initially what we do is like we going to set up a quality target product profile, which we call it like QTTP. So how do we define that? We define that based on the mechanism of action, efficacy, and safety, a dosage form, admin routine, desired quality attribute, as well as regulatory requirements. So we take into a lot of factors to really define that quality target product profile. Once we have that quality target product profile generated based on all these parameters, we create a list of like quality attributes. So once we identify the quality attributes, we rank these quality attributes based on the severity as well as like the risk assessment, like um, how much is it like to understand this each quality attribute in such a way that it plays a big role in patient safety as well as like in the product stability. And once we rank them out, we kind of generate this quality control strategy for each of it, like what is that specific limit or the range this quality attribute will be like really valid for the product. And once we define that, we go ahead and like conduct experiments and really understand, keep on iterating the process in such a way that like we always go back to the scientific literature, platform knowledge, and product understanding and process understanding, what the risks that can be generated for this CQAs in such a way that our product is kind of like operating in the specific immunogenicity as well as like the stability aspects. And once we understand those, once we iterate this process, we have a final CQA identification. So this is a huge process where people do a lot of like brainstorming to understand CQAs for very specific each product. So moving on, so we ideally can talk critical quality attributes based on an mRNA manufacturing point of view, or an mRNA a molecule point of view. These can be subdivided into attribute classes, and this table actually defines an idea of like what the attribute class and what test methods are applied, and what is that quality attribute you're really identifying for this specific mRNA molecule. So the first one, the highlighted ones are the ones I'm going to talk extensively regarding the LC methods, but pretty much like these all critical quality attributes like classes are pretty much important for any drug molecule. So to talk about identity and content, so the test methods people use are PCR and sequencing. So the ideal quality attribute, what we are really understanding is the mRNA sequence, what we anticipated is to generated from a DNA molecule. Is that the correct sequence or not? Ideally, we can use a PCR as sequencing to identify that. And it's also important that like if what is the amount of concentration of RNA that's been produced in such a way that if you have a very efficient process, you have a good yield, and that really helps out to generate like more easily of a drug product. The integrity is the factor. Integrity is primarily comes from, as I said, I showed you like how in vitro transcription happens. You have a lot of product, you have a lot of reagents that interact with each other to produce this mRNA. So it's very important to understand like what is this purity of mRNA that's being generated. So it's a very important quality attribute where you need to understand how much pure is your mRNA that's completely produced. So once that's been produced, and normally we use like ion pair reverse phase high performance liquid chromatography as to understand this mRNA purity. This has been like a method that people have been really developing recently, and when I talk more about like different examples, you'll understand better. And then the other important thing is like, as I said, there are a lot of impurities coming. You need to understand these different amount of impurities as well as the ideal integrity and purity, which I talked before, but they are talking from mRNA molecule itself regarding the different regions of this mRNA molecule plays a very important role regarding translation. So we need to understand purity from that point. Even if you have a whole sequence of mRNA, that doesn't really mean you have your initial phi cap or a poly A in the right way. So these are also very critically important to understand that. So to understand, there are different methods to really look into that, and PCR is one of the methods. Protein-based methods are available because you have reagents like polymerase in it. You can use an ELISA to identify it. So, and there are like LC based methods which pretty much comes for the IP, RP, HPLC, SEC, AEX, and like LCMS methods. The quality attributes we will really identify here would be the phi cap efficiency, poly A, and then aggregation percentage fragment of mRNA, how much is your full percentage to a truncated, and residual DNA, how much is your plasmid DNA still present in your product, and residual NTPs, and the capping reagent. So again, we'll talk more like very specifically for each of them. Then when you look at like safety, this is a pretty compendial methods people use for sterility, endotoxin, and mycoplasma. And potency is mostly based on PCR or bioassay, a cell-based assay for biological activity. It's very product based, and product quality is more like appearance and pH. It's more like a physical methods we apply. So I am going to talk more about like the integrity of like and give an example of how these methods have become more important. LC based methods have become more important. So to start off with, like RNA integrity is a very important factor. So the attribute itself is what is the parameter that really defines here. So the RNA integrity is ideally you're looking at the full length RNA, the desired length product, and how much of this impurity can contribute to immunological responses. So really understanding what is that impurities that are coming with this full length integrated product. So normally the assay options we have is like a quick and user friendly people use is a capillary electrophoresis. So it's the parameters which people have applied across is like it has a high resolution and separation for larger RNAs, which ideally have like more than 1,000 nucleotides, is much uh, easy to run on the capillary electrophoresis. And a separation between 200 to 6,000 is kind of like a very good separation based on the standard curves were produced for this capillary electrophoresis. The biggest challenge is like longer run times and lower repeatability. It all depends on if you have a very good quality of RNA, and like the repeatability is very challenging to get the same data every time. So to really mitigate this and really have like a broader, um, again, this is again a denaturing method where you really denature the RNA to do this. So normally one of the methods we have is kind of like the SAX P800 plus is kind of like that's the one utilized. And I talked a little bit before on this LC methods. So the ion pair reverse phase HPLC method has been like a workhorse nowadays to really identify the RNA integrity. So the separation actually is achieved by an ion pair agent and additives that actually interact with the charged phosphate backbone of RNA. So the phosphate backbone, and it's like really having a native RNA molecule, not denaturing it on NLC method. And people have used polymeric columns, polystyrene, divinyl benzene is one of the columns that people have used, and which is like non-porous and porous, both have been used for this method. And another column people have used is alkylated PSDSD good, which is kind of like good for RNAs that are like lower than 1,000 nucleotides. And silica columns are also good. Normally they have a good resolution between our single stranded RNA compared to a double stranded RNA, so that's where like they play a big role. I'm going to talk like an example of this ion pair reverse phase HPLC in the next slide. I will give an example of a nice study that recently came out, and how did they approach it. The pros are for this method is like shorter run times and high resolution. But the challenge is like if you have like nucleotides more than 5,000 nucleotides, the separation becomes very challenging with this method. So the next method people have always used previously was this anion exchange, because you have like a negatively charged backbone and you're really looking at using a positively charged system. So it's based on charge and ionic interactions. The stronger denaturation conditions. So ideally, you will need like denaturation conditions, because here you're ideally pushing for a higher pH or a higher salt concentration to make the interaction happen for this RNA molecule to interact with the column. So there's a possibility that like these denaturation conditions can denature the RNA. So milder conditions have been applied with ion pairing, anion exchange. We call it IPAX methods. Um, but primarily, like most of the time, if you really want to understand process impurities and really if you want to purify this RNA out, so anion exchange has been applied across different, has been like a workhorse most of the time. Going into size exclusion chromatography, so the separation happens based on the hydrodynamic radius, and the recommended pore size is around like 2 microns. As it plays a big role. The people have tested like different sizes of RNAs with different pore sizes, and people have found like size between like 250 Armstrongs to 400 Armstrongs. A column is like the pore size is very ideal for RNA separation in a good way. But again, people have also tested like different ionic strands of mobile phases and dilution to really test on SEC. Most of the time, SEC is applied to really understand this, more of like aggregation. If the mRNA molecule is kind of getting aggregated, that's where like SEC is applied a lot. Then really understand the purity. It's just like to really understand monomer to like a aggregation. That's what it really is. And it's also applied across this characterization of this poly A tail length. If you have like normally the poly A tail length is between like 150 to 250 nucleotides. If you want to understand how much is it, the biggest challenge is like your resolution on an SEC for RNA is very low. So we don't recommend to go with a lower resolution one. The example, I think the study I'm going to show you, I'm going to talk is kind of like, so this is one of the studies where I think the the goal was to really potentially utilize this, ion pair reverse phase HPLC for separation and characterization of large RNA molecules. The key takeaways from this method, from this article, is kind of like they kind of explored like 13 different ion pairing agents with different hydrophobicities and the retention and selectivity for these different RNAs. And they tested different parameters, like they went from temperatures from 45 to 85 degrees and pH from 6 to 8 and concentrations of all these different ion pairing agents from 25 to 200. And the method was developed to separate oligonucleotides from 1 nucleotide to 6,000 nucleotides. I just want to show some of these figures. So ideally, the first graph is kind of like they just take all these different kind of like ion pairing agents and they kind of create this resolution graph, and they have like a threshold at 1.5. And what we're really looking at is like anything around 0 is kind of like they don't really provide any good resolution, but anything above 1.5 with a which has a green color, the ones ion pairing agents have produced a good resolution of the specific RNA molecule. So they were using this specific ribo standard, which runs from 200 to 6,000, and that's what they're using. And they were using this DNA pack RP column to do this, and it's a 20-minute run column, 20-minute total run. So they ran different mobile phases, and what they found was like this isopropyl ethylene was kind of observed to be giving the best resolution. And as they did a lot of next steps, and as they also used like butylenamine, and they tested like different methods, they optimized different methods, and what they came up with like they identified like this IPA of like butylene amine and TPA played a very good role at like 125 millimolar combination. And once they started to play with the reverse phase condition with the acetonitrile, what they observed was like a 20-minute run. They used like a 10 to 100 deoxythymidines and the ribo-ruler high molecular range, which is from this 200 to 6,000. And what they were able to identify in this method was they were able to really specifically segregate these individual molecules on this 20-minute run on the column. And they were using the same column, DNA pack RP column at this point. From there, they optimized this system more, this method more, and they ideally added another column with the DNA pack RP column. They added an SCC column next to it. And then they were able to modify the method with more gradients. Still, the method is like 20 minutes. And then they were able to show from single nucleotides to like a 6,000 nucleotides. So this is a great example of how RNA integrity can be identified, and like this is one of the examples where this group was really showing how ion pair reverse phase liquid chromatography can be applied to identify RNA integrity. It's a very good paper, like to understand all these applications. So to talk about next, like after RNA integrity, with the RNA integrity, I also want to talk about this phi cap and the phi cap capping efficiency. As I said, it's very important for stability and translation. So in ideal mammalian phi cap structures consists of it consists of a methylated at its N7 position and to the link of the triphosphate linkage. So to make like the similar mammalian phi cap structure, the synthetic RNAs also were like modified across that like the N7 position with a methyl group. And people also identified that like this is a cap 0 position. And once people also identified that like you can also modify, which is like the R1 and the R2 positions with methyl in such a way that like you can the methylation of this cap region would help for the stability of the RNA molecule. So that's what, if the cap 1 region has the methyl, it's called as a cap 1, and the R2 has like it's called cap 2. So because like you have like a methyl at the N region and the R2 region is like 2 methyl, so it's like cap 2. That's what they call it. So the analysis of RNA purity actually normally happens with the phi cap and the phi capping efficiency for the LCMS method. So in general, you kind of take the RNA molecule and then you use like a RNA-T or an RNA-H and digestion. Once you digest all these molecules, you ideally use an LC method to segregate them out. And then the one way to really understand is kind of, you can understand like how many of your phi cap compared to the uncapped regions, and then you get an efficiency of uncapping. Or then ideally you collect everything, and then you can ideally go for LCMS, where you ideally understand the mass by charge ratio, and then go identify each nucleotide from there. So moving on, So the different kind of LC based methods applied are primarily based on the enzyme that we use for breaking down the RNA molecule to identify the integrity of the phi cap and the phi capping efficiency. So it ideally provides more information about the translational efficacy and the RNA stability. So people have used a lot of RNA-H, which has been recommended um, uh, by most of the regulatory groups. So it's an endonuclease, cleaves the diphosphate bond of RNA DNA hybrids, and it does not cleave any double-stranded RNA. So if any DNA is involved, it actually breaks down. Um, so so it's mostly like it it helps out. Like one of the pros of this is like it's a label free application where you kind of like you're just taking the RNA and you're just like breaking it and really go use an LCMS method. Normally a C18 column is used with an RP trap analyzer for an LC-MS, and then you identify the specific fragments. The biggest challenge with RNA-H method is like it's a low recovery and more optimization might be required for your specific application. So it takes a little bit of time to really, get the method up and really setting up. Then there's another enzyme which is the RNase-T enzyme, which ideally it's an endonuclease. It just generates like a cut around like ending at G. So one of the great things about RNST is like it's not going to cut any of your poly A because like you don't have any G in there. So this is also an enzyme that's used for poly A. The high resolution, this enzyme is kind of like utilized, but like the biggest challenge is you might need a high resolution accurate mass approach, which is kind of like you might have to have like a template or a control, which you ideally have to have in the method to see that the RNA-T is working in the right way, and then you compare your control with your test molecule. Again, the LCMS process is again using a C18 column with an RB trap analyzer. There's another nuclease, which is nuclease P1. So this ideally generates like intact phi dash caps with methyl groups. So it's ideally tested out when if you have an RNA molecule generated, you want to add a phi cap later on, and then you really want to check like if that really happened, you can use this nucleus P and really identify this N7 methylation. Did it happen or not with this nucleus P? So one of the important things with nucleus P is kind of like it can be applied in a systematic way. You can ideally use nucleus P digestion and then go ahead and do your RNase-H or RNase-T digestion. It's a sequential thing. It's not going to interfere with other enzymatic steps. Again, it's an LCMS based method, and you have like more like a C18 column and RP trap. So talking about more from a poly A tail and heterogeneity. So poly A tail is pretty much for RNA stability and protects RNA from degradation. It's typically in the range of like 100 to 150 nucleotides. And the heterogeneity in poly A tail normally arises from this transcriptional, because you have poly A available, like this transcriptional slippage can happen where ideally there can be a base that can be introduced into the poly A. As I said before, the RNase-T is one of the method people use because it only generates a G, so you're pretty much saving your whole poly A. That's the easiest way, and people have used OLE-DT purification to get these tails off, and then we ideally use this reverse phase MS analysis on a C18 column. It's a label-free method, and people also have, if you don't have a mass analyzer, you want to go with an LC and you want to go with a UV. And people have developed methods with an SCC, which ideally you test with the 250 to 400 Armstrong columns with a 0.1 phosphate buffer. And you might need more optimization because you might require like standards to establish this method. But yeah, it is right now it's pretty much like poly A. It's like easy. There are like more methods developing, but like right now we have these two methods available. So, talking from a critical quality attributes point of view, from an mRNA therapeutic point of view, so it's like we have limited knowledge and no sufficient accumulated information regarding previous development of similar products. People are developing right now, understanding, developing these methods, and the product development knowledge is kind of still limited, I would say, and regulatory guidance is still evolving on these. So regulatory guidance based on agencies, you have like the European agency and the FDA also produce few of them, and non-agency based or like from UPS and the biopharma, they have like a pretty good kind of like analytical procedure methods. This is just the guidance you can ideally get. Yeah, these are some of the references actually you can refer to, and these are pretty recently published actually what I'm talking about, so you can go visit them and really get an idea like how the field itself from in liquid chromatography is moving towards understanding these large RNA molecules. Yeah. Yeah, I'd say acknowledgements would be like, yeah, these are my acknowledgements, and like I would say thank you very much for your time, and I will definitely have any questions